Or brainstem, down the descending tracts, right, into the gray matter, out through the anterior horn, which is the motor root, right, out to the spinal nerve, to the ventral rami, to the trunks, to the cords, to the peripheral nerves, smaller and smaller nerves, ultimately get to the neuromuscular junction. It's the point of contact, or not really contact, not really contact, because that's a bad word, because in the synaptic cleft you don't have contact, you have a very small gap in the cleft. But it's the last neuron that connects with the first muscle cell, right? So if we look at our involve, right, you know, we have the synaptic vesicles, and here's the muscle, this is the neuron, right? We know the action potential in the neuron, right, that occurs in the dendrite. The sodium channels open up, right? We have depolarization. The potassium channels open up. We have repolarization. We have everything coming down through the axon, right? The calcium channels here open up. They force the permeability of the synaptic vesicles, right, through the protein synaptotegument. Yep, synaptotegument forces the synaptic vesicles to release the neurotransmitter, right? Right, that's the neuron 1. This is neuron number 2. Actually, could have been neuron number 2000. Okay, but it's the final neuron. This is the first muscle cell, number 1. Okay. And what happens? What's the neurotransmitter of choice?
Acetylcholine proteins.
Acetylcholine, right? And what are these?
Intermembrane protein.
A whole bunch of ACH receptors on the sarcolemma. The sarcolemma is the cell membrane of the muscle cell, right? Or the fiber. The muscle cell, which is over here. This is a muscle cell here, right? In here. The muscle fiber, the cell. On the cell membrane of that muscle cell are acetylcholine receptors. When the action potential comes down, changes the permeability of the synaptic vesicles, they bump up against the N-terminal bulbs, spew out their neurotransmitter into the cleft, right? So no contact, right? I use the term contact. That was not contact. That was no good, because we know that there is no direct contact. This is in the synaptic cleft. The neurotransmitter comes across, binds to the acetylcholine receptors. Okay, so that's where we left off when we talked about the nervous system. So now, what happens is this: the acetylcholine receptors cause a series of biochemical reactions so that the sarcoplasmic reticulum also referred to sometimes as the SER, the sarcoplasmic reticulum, which is the endoplasmic reticulum in the muscle cell. The sarcoplasmic reticulum releases calcium. So the permeability of the sarcoplasmic reticulum is changed so that calcium is released. Calcium is stored in the sarcoplasmic reticulum. And the acetylcholine binding to the acetylcholine receptors causes a change in the permeability of the membrane of the sarcoplasmic reticulum, which releases a lot of calcium into the sarcoplasmic reticulum. Now, in the muscle cells, there is a whole series of other vessels or other ways which things get around, and those are called the T-tubules. So these yellow things here on the model that we have, the yellow sort of the network here in the middle, these are the sarcoplasmic reticulum. They're filled with calcium. These larger yellow areas here are called the T-tubules. And the T-tubules also have a lot of calcium. Okay? So once the acetylcholine binds to the acetylcholine receptors, it changes the permeability of the sarcoplasmic reticulum, and it releases the calcium into the muscle cell. Now, what this calcium does is, the incremental calcium will bind
with axon of the
calcium is increased by the acetylcholine binding to the acetylcholine receptors. So the sarcoplasmic reticulum releases calcium, and that extra calcium in the system changes the conformational shape of troponin. Remember our old friend here, the blocking protein? What's holding up contraction? What's holding up contraction is the troponin tropomyosin complex sitting on the actin binding sites, right? We have to find some way to release that troponin tropomyosin complex to allow what? Allow the myosin heads to flip up and bind on the actin binding site so that we can complete contraction.
so it's calcium that
So it's the calcium that
does
binds
the
to
tricks.
the troponin, which is part of this troponin tropomyosin complex. When it binds to the troponin changes its shape or its structure, changes its morphology. So when it changes its shape, it literally cannot fit on the actin binding site anymore, and change of the conformational shape is what releases the troponin tropomyosin complex from the actin binding site. So we have a release
calcium that causes
of the
Things
complex
to remove, change the shape so,
from the actin binding site
so they can fit.
And once this release occurs, then we're back to what we began talking about, right? Once the troponin tropomyosin complex is taken off the actin binding sites, then we can have the myosin heads click on to the actin binding sites. Once that occurs, we have the formation of the cross bridge, and then ultimately the power stroke goes 45 degrees left and right to 45 degrees left and right. And we have a shortening of the sarcomere. Okay. So what we have then is a combination effect between the nervous system, right, where the, say I want to move my hand or I want to contract my fist. Okay, so I'm going to punch somebody out. I want to contract my fist. So my brain makes that decision in my association neurons, travels down the brain stem through a descending tract, right, goes over into, it's in my arm, I'm going to contract my fist, so it's got to come out somewhere in this area here, hops over into the gray matter through the anterior horn, right, the motor root, comes out through the spinal nerve into the ventral rami, into the cord, the plexuses, the individual nerves going down to my fist, where then ultimately, at the last neuron, my muscles in my fist are excited, right? One of the characteristics of muscle tissue was its ability to become excited or to become depolarized, to continue and act, to carry out an action potential. So then the synaptic vesicles in those neurons that are impinging on the muscles in my hand spill out the acetylcholine. They bind to the acetylcholine receptors. The acetylcholine receptors release the calcium from the sarcoplasmic reticulum. Actually goes through the T tubules as well, and then will attach to the blocking protein that up until when my hand is relaxed, we know that those blocking proteins are on the actin binding site, so my hand is not contracted. When I contract my fist, we know that decision has been made, the acetylcholine has bound to the acetylcholine receptors, calcium has been released, calcium has changed the conformational shape of the troponin tropomyosin complex. Because it changes shape, it can no longer sit on that site. It's a very conformational process. It's a very structural process, just like a receptor is. Fits in like a puzzle, like a piece of a puzzle. So when that is lifted off, then we have the formation of the heads, and then those heads swivel, and ultimately that causes contraction. Yes?
No. They were like, why is it so high?
What's that?
They asked me why was it so high.
Oh, you were... Yes.
Sorry, can you explain it? Can you just explain that again?
That's that again.
So we're going to go over it all again on Wednesday, but I wanted you to at least be exposed to it, like the active binding sites to be exposed. So I'd like you to have you read it by Wednesday, because we're going to go over it all again. But this is the most important part of the physiology in terms of muscle contraction. Okay, so what we're trying to do here is integrate what we've learned in the nervous system, where we kind of left off here, didn't we? I mean, if you look back at what we talked about, we kind of left a dead end there, didn't we? Okay, so now we're going to sort of complete the process, because the process ultimately, when this contracts, right, it contracts the tendon and the bone. When the muscle contracts, it moves the bone. So we need to have this piece between the skeletal system and the nervous system. Okay? So the command, the motor roots, the commands, remember this is all motor now. Okay? It's not sensory, it's not going up to the brain. So all the decisions are motor in terms of contraction of the muscles. So this is a motor response from the brain to the cord, to the anterior ventral roots, out through the spinal nerve, out through the ventral rami, out through the cords and the roots and the plexuses, ultimately to the individual peripheral nerves, the individual neurons. Then we have the action of the muscle. How does the muscle contract? Well, it contracts because of the signaling mechanism from the nervous system. And that is done by the neurotransmitter acetylcholine. Once it binds to the acetylcholine receptors, that then opens up the calcium channels in the sarcoplasmic reticulum. Calcium floods the system. All those calcium molecules jump onto these blocking proteins, and it changes the shape. So when it changes the shape of those blocking proteins, then they're lifted off because they don't fit anymore. It'd be like changing the piece of a puzzle. The puzzle doesn't fit anymore, and so it can't sit on it can't block these sites. So as soon as that happens and the blocking proteins are released, the myosin heads are very opportunistic, and they immediately flick onto those binding sites because they have an affinity for them. They have an attraction to those binding sites. And once they flip on, formation of the cross bridges and then the power stroke, and that's what causes contraction. And then ultimately, after a period of refractory period of time, then the calcium will lift off, and then as soon as that does, the troponin comes back on and blocks the sites, and then we have relaxation.